alaikum wa rahmatullahi wa barakatuh. Today I want to share with you some knowledge about the genetic and inheritance of a blood group system. So this lectures actually I gave it to my student, but I have a second thought why I didn't share the knowledge with everyone who can benefit from it. So the lecture objective was by the end of this lectures, my student will be able to explain the genetic basis of the blood group antigen and inheritance patterns, differentiate between a phenotype, genotype, understanding the effect of zygosity, recognize how genetic variations lead to rare or null phenotype and autoantibody formation, and at the end apply the genetic principle to ensure safe transfusion, which is very important in the main goal for the transfusion medicine, and interpret clinical relevant. So why genetic matter and the immunohematology? I hope at the end of this lectures you will be able to answer these questions very easily. So let's now concentrate on the corner on the right, the image on the right. As you can see here, there is a double strand of the DNA weave around each other and wrap around a protein called histone. This is to be able to fit inside the cell because the DNA is very large, as you know. When the histone and the DNA wrap together, they organize on the shape of the chromatin. And as you can see, I hope the arrow is appear here. As you can see here, one chromatin come from mom and one chromatin come from dad, and they join together at the centers by something called the centromere. After that, the chromosomes fit inside the nucleus, and we will see more detail about the chromosomes in the next few slides. So let's read together. The genetic information is carried on the double-stranded DNA, the genetic material, which is necessary to maintain the life of the cells and the organism. The DNA, as I said, is packed and organized on structures called chromosomes. These chromosomes carry the gene that determine our trait, including a blood group antigen. And this is logical because, as I said, one chromatin from mom come with all the genes on it, and one from the dad come from all the gene on it. So these two chromatin totally called chromosomes determine our trait, including a blood group antigen. So a human cell have 46 chromosomes, 23 pair, one from mom and one from dad, come together in a pair, as you can see here on the right pictures. Each chromatin colored with blue. Two chromatin form one chromosomes. 22 pair are autosomal, carry the same kind of the gene. So the gene come from mom the same as the gene come from dad. Because of this, we called it autosomal chromosomes. Autosomal chromosomes. One pair of the 6 chromosomes determine the sex. So on the female, one X come from mom, one X come from dad. For the male, one X come from mom, Y come from dad always. So no male can inherit the X from dad. They always inherit the Y from dad, and this is a very important things to understand, because when we go to the sex-linked inheritance, you will apply this knowledge. The chromosome structures. as you can see here on the right image, each chromosome has two sections. The top section, we called it the short arm, and the lower section, we called it the short arm at the top and the long arm at the bottom. And they joined from the by something called the centromere. It's not always in the center, by the way. Here, the gene mapping. As you can see here, the pictures on the right corner, this is a chromatin, and by the specific stain, they stain it to give a look of the white and black bands. Each band, we called it band or region, and this region we can sub-classify it or reclassify it again to have the subband or subregion, as you can see here on the lower image. So why this is important? And the gene mapping chromosomes serve as the road map showing the relative positions of the gene to understand the inheritance and locate a trait or diseases. Numbering system, the number follow the P or Q indicate the band region or subregion. Numbering start from the centromere and move outward toward the telomere. I hope the arrow is appear now. So the numbering start from the centromere up and from the centromere down. For example, if I said I have a blood group system called MAL, the gene encoding this blood group located in the 2q11.1. What does this mean? If you read it on the textbook, that means the gene located in chromosomes 2, long arm, region 11, and the subregion 1. So now you are familiar. Go to the or look at the image on the right corner. Now you are familiar with the chromosomes. It's two chromatin joined by the centromere. And if my gene of interest located in this area, we called it locus. If I want to enlarge this area, I can see here a stretch of the DNA. And it contains more than one gene, of course. I'm interested in gene number 2. I can, for example, if I want to look to the sequence, exact sequence of this gene, I can use molecular testing like the PCR followed by the Sanger sequencing, and I can identify the exact sequence of this gene. So the gene is the segment of the DNA that occupied a specific location on a chromosomes known as a gene locus. For the pictures down, I want to give you a few review about what happened from the gene until I get the protein. So in the pictures, the gene, it consists of the promoter and regulatory genes, exon, intron, but this is in the DNA. The DNA cannot exit the nucleus. So if I need the protein, there is a copying process, a transcription, converted the DNA or copy the DNA, the part of the DNA to be RNA. So the RNA consists of the intron, exon. By splicing a process, I get only the exon, and we called it messenger RNA. The messenger RNA exits the nucleus to the endoplasmic reticulum, and to be specific, until it's reached the ribosomes. On the ribosomes, it converted the nucleotide and the RNA to the amino acid chain, a linear protein. Then the linear protein, it travels through the rough endoplasmic reticulum, and through the journey it gets folded, and transport to the Golgi apparatus, where they decorate this protein. For example, they add the sugar, and we call this a process glycosylation. Then the protein is ready, either exported and anchored on the cell membrane, or secreted as a secretory protein, or can go to the other organelles inside the cell. So the gene encode the protein, structural or the enzymatic. So on the blood group area, the gene encode the protein, or the protein that carry the blood group antigen, or they encode the enzyme that help to synthesize the blood group antigen. In both way, we call them a carrier, which are expressed on the red cell's membrane and known as the carrier. So if the gene encode the protein, it will be on the cell membrane, carry the antigen. If it's encode the enzyme, it helps to synthesize the carbohydrate on the top of the protein or on the top of the lipid, and it carry the blood group antigen. So the antigen, the blood group marker, carried on this carrier. As you can see here on the right corner, the red blood cells, and there is multi carriers on it. And if we take a Duffy blood group system, the Duffy protein, and enlarge it on the below image, this is how it's look on the surface of the red blood cell. As you can see, it's the carrier and it's carry a protein. So for example, in 42 position, there is amino acid, if it's Duffy A or Duffy B, or it can be here the Duffy 3, and so on. So the main point to understand here, the red blood cells carry multiple protein, glycoprotein, glycolipid, that carry a blood group antigen. And we can see here the antigen are carried on this carrier either on the form of protein structures, carbohydrate epitope synthesized by the gene encoding enzyme, which the antibody can recognize. And this is a very important thing. So for example, if there is a change on the DNA sequence that affect on the protein structures and affect on the protein expression, this change allows the immune system to be recognized as a foreign and develop the antibody. If the patient develop antibody, I can authorize it as a blood group antigen. But if the patient do not develop any antibody against this antigen, we cannot call it a blood group antigen. This is a very important concept. So the presence or absence of a blood group antigen is under the genetic control, and the identity of the blood group antigen defined by the development of the antibody. So the blood group genetic, it's the gene encoding the carrier. So the carrier protein, glycoprotein, glycolipid that carry the antigen. So I think I can stop here and continue on the second part of this video.